Bismillahirrahmanirrahim. I am Dr. Muhammad Tayyab, serving as Associate Professor in the Institute of Biochemistry and Biotechnology, University of Veterinary and Animal Sciences, Lahore. So, I am here with the title Locally Characterized Recombinant Enzymatic Mixture, a step for uplift of poultry feed industry of Pakistan. So, basically, there are two important aspects in this talk. Number one, that is the poultry feed industry, and number two, that is recombinant DNA technology. So, let's talk about these one by one. So, first of all, poultry feed industry. Poultry feed industry is actually an important industry of Pakistan that is committed to provide good value, good quality meat to the nation. So this industry is actually facing some sort of issues. So what sort of issues? Like the feed cost, that is the important aspect. And feed contributes approximately 77% of total cost of the poultry. So that means we have to discuss the poultry feed. And regarding the poultry feed, basically there are two possibilities, like we can go to animal-based feed or we can go to plant-based feed. In case of animal-based feed, that is quite expensive. And while in case of plant-based feed, the raw materials are cheaper. So when we are going to utilize the plant-based feed, then we have to face the problem like plant-based feed that contain a huge quantity of cellulose, xylan, proteins, and phytate. These are the compounds; those are not being digested completely in the body of the poultry bird. That is might be due to the low production of enzyme or unavailability of enzyme, and its solution is to supplement the feed with the enzymes, because supplementation of feed with the enzymes will result in the complete digestion of these compounds, and these will provide an extra source of energy, because in the absence of enzyme, the materials like cellulose, xylan, proteins, these are not being digested and simply pass through the digestive tract with the manure from the body. And secondly, the supplementation of feed with the enzyme that is environment friendly, that will improve the efficacy of feed utilization and improve the poultry bird growth. Thirdly, this will save the huge foreign exchange, which is actually being invested on the on the on getting the enzymes. So this means enzyme production that is actually the solution for the complete digestion of poultry feed, and this will actually put a positive impact on the poultry feed industry. So now next question is that which sort of enzymes will be suitable for this industry? Actually, we need thermostable enzymes. Thermostable enzymes are the enzymes which will be stable at a high temperature, which can withstand a high temperature, because the pelletization step during the feed preparation that requires the treatment of feed at high temperature. Then these enzymes should be able to work in the body of the poultry bird. And so here in this study, the enzymes being produced, that is actually from Thermatoga naphthophila, that is actually a hyperthermophile. This is actually the overall strategy of working, and actually the work was started with the availability of genomic DNA of Thermatoga naphthophila. So specific genes from this genome will be amplified by the PCR so that we can have multiple copies. Then the cloning of gene of interest to cloning vector, and then shifting of that and or subcloning to expression vector. And after cloning to the expression vector, you can have the process of transformation of E. coli BL21 codon plus cells that will be utilized for the production of the specific enzymes. Then the utilization of produced protein for the purification and for further characterization, and ultimately for the utilization of these enzymes for poultry trials. So this was the overall strategy, and as a result, you can see actually the results. These are being discussed briefly. So first enzyme was phytase, which was responsible for the breakdown of phytate to its basic components, because phytate can work as an anti-nutritional factor. So the SPS page analysis, which shows the purification of this phytase, then its impact of temperature shows the optimal activity at 80 degrees Celsius. In the same way, you can see this enzyme was thermostable. Optimal pH was between 6 to 7. Then iron was actually required for the activity of this enzyme, and then the kinetic studies. Secondly, the cellulase, that is also an important enzyme which is responsible for the breakdown of cellulose to its simple basic monomeric units. So the SPS page, which shows the purification of this enzyme, this is actually a 39 kDa protein. Then optimal temperature, which was found 90 degrees Celsius, and the thermostability studies. The pH 4 is required for the optimal activity of this enzyme. Effect of metalines, detergents, and kinetic studies, that was also done for the activity of this enzyme. That is, next is cellulase. The cellulase was actually purified, and it was found to be a 36 kDa protein. Optimal temperature, pH, metalines impact, detergent impact, this was also examined, and then finally the kinetic studies of this enzyme was observed. Then protease. This protease was actually of about 36 kDa protein. Here 60 degrees Celsius was actually the optimal temperature. pH 9 was actually the optimal pH. Then effect of metalines, detergents, and the kinetic studies, these experiments were performed. So after having the characterization of these enzymes was utilized for the actually supplementation of poultry feed, and feed was utilized for poultry trials, and the output of trials that concluded these enzymes actually showed a positive impact on the growth of the poultry bird. So finally, you can actually conclude that supplementation of poultry feed with locally produced enzymes, these showed a significant impact on the weight gain of the poultry birds. Thus, locally produced enzymes will be utilized as a mixture for the supplementation of industrial demands of enzymes, and this will save the huge foreign exchange for the import of these enzymes in future. Thank you. If you have some question, you can ask.